Sahiba will be speaking today on Euclidating the role of ATP-IF1 in mitophagy and mitochondrial biogenesis. Thank you for your time, Sahiba, and I can't wait to hear about your work that you did this summer.
Hello, everyone. Thank you, Lily, for that really sweet introduction. My name is Sahiba, and I was a summer intern at Sense Research Foundation working with the MitoSense team, and I'm thrilled to be talking about my research, which is focusing on elucidating the role of ATP-IF1 in mitophagy and mitochondrial biogenesis. So, mitochondria are the powerhouse of the cell, right? But how often have we given credit to the millions of generators, rotors, and engines working within that powerhouse, working to give us energy? And most important, what if these generators stopped working? Over 1 in 5,000 people in the US suffer from mitochondrial diseases without any cure, and many more are affected by mitochondrial dysfunction in age related diseases like Alzheimer's and Parkinson's. Dysfunctional mitochondria are heteroplasmic, meaning that they contain a mixture of mutated mitochondrial DNA, or mtDNA, and normal mtDNA. As the level of heteroplasmy increases beyond a certain threshold, disease phenotypes become present. Therefore, by selectively eliminating mutated mtDNA, it offers a promising approach to therapeutics for mitochondrial disease. Our lab in particular seeks to use the cell's mitochondrial destruction system, also known as mitophagy, to selectively eliminate mutated mtDNA and improve mitochondrial function. So, mitophagy can be induced by changes in the inner mitochondrial membrane, or IMM potential, which is made up of a gradient of hydrogen ions. The gradient is regulated by a protein complex in the IMM called ATPase complex 5. This protein complex has a synthase and hydrolase component. Its synthase component makes ATP by using the passive flow of hydrogen ions down their concentration gradient. The hydrolase component is vital to maintaining the IMM potential by pumping these hydrogen ions against their gradient. So without this hydrolase component, a stressed or dysfunctional mitochondria would not be able to stabilize itself and would undergo mitophagy. However, healthy mitochondria would still be able to function without the stabilizing properties because they would rarely be destabilized in the first place. So the protein we're looking at, ATP-IF1, inhibits this hydrolase component of ATPase complex 5. So first, that ATP-IF1 gene is expressed, which increases the production of the ATP-IF1 protein, also known as IF1 for short. Once it's produced, it dimerizes and binds to ATP synthase, which inhibits that hydrolase function. And then the IMM potential cannot be modulated, leading to a decrease in that potential. And that further leads to PINK1 and Parkin mitophagy pathways, and finally mitophagy. In the diagram shown, the two mitochondria have been destabilized by a membrane uncoupler called CCCP. When ATP-IF1 is present, it inhibits that hydrolase function, and as you can see, all the H plus ions are stuck on the inside, leading to an imbalance and ultimately mitophagy. In an absence of ATP-IF1, that hydrolase function can stabilize the cell and mitophagy won't happen. So we hypothesize that by overexpressing ATP-IF1, we will selectively induce mitophagy in dysfunctional cells, leaving those functional mitochondria unharmed. And to investigate this hypothesis, we grew out 5 stable cell lines. So our 2 controls are our 143b wild-type cancer cells and BH2 heteroplasmic cells with 6 mitochondrial gene deletions, which lead to deficiencies in oxidative phosphorylation, or the machinery in which ATP is produced. So our 3 ATP-IF1 constructs are OE, which contains a plasmid that overexpresses the ATP-IF1 gene, S39A, which contains a dephosphorylated active version of ATP-IF1, and S39E, which contains the phosphorylated and inactivated protein of ATP-IF1. And we'll be using analysis at the genomic, RNA, and functional level. qPCR is our measure of mutated mtDNA load. RT-qPCR is our measures of the levels of ATPIF1 in mRNA form, which we use to confirm that our plasmids are expressing the gene. And then we also had functional assays to show whether the mitochondria can actually improve their function after those dysfunctional mitochondria have been cleared. So after each successive round of mitophagy, which takes about 2 weeks, we hope to see that the mutated mtDNA load decreases and that our cells show improved oxidative phosphorylation functioning in the OE and S39 cell lines, again, because we've gotten rid of those dysfunctional mitochondria through mitophagy, leaving only the functional ones behind. However, this is not what we saw. So after 4 weeks on doxycycline, which activates all of the plasmids in our cells, the constructs had the same mutated mtDNA load as our diseased control. Further, in an impedance assay, the cells could not grow on galactose, which can only be metabolized in oxidative phosphorylation. And since those cells could not grow in galactose, oxidative phosphorylation is dysfunctional. And so, in an effort to troubleshoot, we referred to an article that analyzed the effect of ATPIF1 overexpression in fruit flies. So the authors, Kendall et al., needed to use ATPIF1 in conjunction with an MF1 inhibitor to see an effect. So as you can see in the graph below, and specifically the region that I highlighted with a black box, the mutated mtDNA load is much higher when just expressing ATPIF1 alone when compared to expressing ATPIF1 overexpressed with the MFN knockout. So what is MFN1? It's a gene called mitofusin-1 that is responsible for mitochondrial fusion. Mitochondria are not static organelles. They fuse and they split apart. And if they are stressed or dysfunctional, they can fuse. And that fusion, it actually makes them more resistant to mitophagy and prevents ATPIF1 from doing its job to get rid of those dysfunctional mitochondria. And so we obtained a fusion inhibitor called MFI8 to test its ability to induce mitophagy when expressed with ATPIF1. We also obtained a fission inhibitor called MDV1, or diamine-related protein 1, to observe that the effects that we're seeing are related to fusion and fission only and not any other process. And finally, we have DMSO, which is our negative control. And so after treating for a certain amount of time that we found according to the literature, we used JC1 flow cytometry and qPCR to test whether we have functional recovery and reduced mitochondrial mutation loads. So after 24 hours, we saw a surprising result. And just to let you know, so the MFI ratio is showing fluorescence intensity. So mitochondria that stain red with the JC1 dye are healthy, while those that are stained green have dysfunctional IMM potentials. So the higher the MFI ratio, the healthier the mitochondria. And as you can see, the overexpressed cells in MFI8 treatment had a significant jump in that ratio just after 24 hours, and this seemed to offer a promising hope that MFI8 with the OE construct can improve mitochondrial health. However, 1 week after treatment, that effect disappeared. As you can see, the MFI ratios in all of the MFI treatments have about leveled out. And further, we also looked at our mutated mtDNA load, and again, all of the constructs have about the same levels. There's no difference that would be physiologically significant. So once again, to kind of troubleshoot, we tried to perturb our dosage and timing of MFI8 treatments to try and enhance that effect that we saw with overexpressed after 24 hours and try to maintain that for longer to see if that could affect our mutated mtDNA load. And so we had 3 different timelines. The first was an MFI8 treatment that was done every about 2 days for 6 hours. And then we also had MFI8 that was treated for 24 hours and then not anymore after that. And then our DMSO control was also for 24 hours. And so As you can see in the JC1 flow cytometry, again, the constructs are about the same with not any major differences between the constructs. However, we do see something interesting in our qPCR data that shows mutated mtDNA load. The mutated mtDNA load is higher in the DMSO controls compared to all the constructs in MFI8. And so this seems to point to the fact that MFI8 actually works independently of ATPIF1, and it seems to drive membrane potential changes upstream of ATPIF1. So therefore, we say that ATPIF1 cannot drive mitophagy of dysfunctional mitochondria alone. Therapeutic development, therefore, needs to consider the influence of both biogenesis and mitophagy. Future possibilities also include investigating another part of the ATPIF1 pathway, particularly the PINK1 and Parkin genes and IMM potential changes. We can also analyze upstream regulators of ATPIF1 to deduce the exact role of ATPIF1 in mitophagy. For example, this could include looking at ATPIF1 kinases and regulators of mitochondrial fusion and fission. And so as we unravel these links between the mitochondrial engines, rotors, and generators, and also fusion, fission, and mitophagy, we come closer to finding a cure for mitochondrial dysfunction. And before I end, I would like to give a huge thank you to the entire Mitosense team, including Dr. Kate, Dr. Bhumanathan, Pavna, Annika, and Jonathan, and the Apoptosense team, Dr. Tess, Dr. Yafe, and Gabriel for their help with flow cytometry, and the Sense Education Department, including Dr. Lily and Christine, for their this wonderful opportunity. Thank you.
Thank you so much, Sahiba, and for your research and for all your hard work this summer. I'm going to throw out some questions from the chat. So Michael Ray has asked you to translate this. Is there a suitable small molecule mitofusin 1 inhibitor?
That's a good question. So mitofusin 1 is actually not looked at in vivo because of other effects that it could have. So it wouldn't be a suitable therapy, but there are definitely other therapies that are being developed to hopefully, indirectly target the fusion and fission genes.
Great to hear. Are there any other questions either I guess they have to be in the chat, but I'll give everyone one more second to respond because I see one more coming in from Michael Ray. So we'll give it one more second. Is there any reason to think that this would not work as well in quiescent or post-mitotic cells?
I think it should not make a difference. However, I'm not certain about the answer, and I would have to do a little bit more research and get back to you on that. But it's
You'd have to
definitely
come back for another term with us then.
yeah.
In that scenario. Oh, I see another question coming in. Sorry. Waiting for the typers out there. Michael gave you a thumbs up. We'll take this one last question and then we'll head on to our next speaker. But Gary Abramson asked, would you like to pursue a career in rejuvenation biotechnology? Which I think is a great question. What's your future plans, Sahiba?
Yeah, I'm definitely open to it. I had a great time this summer, and there's so much within this field that I would love to explore it more. And absolutely, thank you so much.
That's fantastic to hear. And we were really delighted to have you with us this summer. So thank you very much. And with that, I will turn it over to Maria.
Wow, that was fantastic, Sahiba. Thank you. We are really proud of our students and the incredible work that Lily is doing with our education department.